Hi, my name is Elizabeth Simcoe, and today's date is November 2nd of 2025. Chapter 32, Nucleotides. This chapter contains 4 sections: the importance of nucleotides, purines and pyrimidines, toxicity in synthetic nucleotide analogs, and DNA-RNA polynucleotides. The learning objectives for this chapter are as follows. Write structural formulas to represent the amino and oxotomers of a purine and of a pyrimidine and state which tautomer predominates under physiological conditions. Reproduce the structural formulas for the principal nucleotides present in DNA and in RNA and the less common nucleotides 5-methylcytosine, 5-hydroxymethylcytosine, and pseudouridine. Represent D-ribose or 2-deoxy-D-ribose, linked as either syn or an anticonformer to a purine. Name the bond between the sugar and the base, and indicate which conformer predominates under most physiologic conditions. Number the C and N atoms of a pyrimidine ribonucleoside and of a purine deoxyribonucleoside, including a primed numeral for C atoms of the sugars. Compare the phosphoryl group transfer potential of each phosphoryl group of a nucleoside triphosphate. Outline the physiologic condition roles for the cyclic phosphodiesters CAMP and CGMP. Appreciate the polynucleotides are directional macromolecules composed of mononucleotides linked by 3' to 5' phosphodiester bonds. Be familiar with the abbreviated representations of polynucleotide structures such as PTPGPT or TGCA-TCA, for which the 5' end is always shown on the left and all phosphodiester bonds are 3' to 5'. For specific synthetic analogs of purine and pyrimidine bases and their derivatives that have served as anti-cancer drugs, indicate in which ways these compounds inhibit metabolism. And then starting with section 1, the importance of nucleotides, we are going to kind of just discuss some of the functions throughout the body. So one of the primary functions of nucleotides is through DNA replication, transcription, and translation. Nucleotides like adenine, guanine, cytosine, and thymine, abbreviated A, G, C, and T, and even uracil, abbreviated U, serve as the genetic code needed for DNA to replicate itself as seen here. Once DNA has successfully copied its genetic code, it can serve as a template to code for messenger RNA, or mRNA, which uses nucleotides to assemble itself as well. After the mRNA has been made, nucleotides are grouped into threes and serve as codons that tell the cell which amino acids are needed to build specific proteins. Another crucial role of nucleotides is the regulation of enzyme activity. Nucleotides such as ADP, ATP, AMP, or CTP play a role in allosteric regulation of enzymes by using feedback loops. Feedback loops can be positive and tell the reaction to speed up and keep going, or negative and tell the reaction to slow down and stop. An example that was covered in another chapter of this class is glycolysis. When hexokinase turns glucose into glucose 6-phosphate, a molecule of ATP is turned into ADP. Consequently, ADP acts as a point of regulation for hexokinase, where if too much ADP is produced, the pathway knows to stop converting glucose to glucose 6-phosphate. Or, if too little ADP is produced, the pathway knows to convert more glucose into glucose 6-phosphate. So here's kind of a visual representative of that feedback loop. Another function of nucleotides is signal transduction. CAMP and CGMP are secondary messengers in hormone transductions, acting as a bridge between extracellular receptors and intracellular target proteins. Additionally, GTP and GDP play a role in G protein-coupled receptors, where GDP inactivates the receptor and GTP activates it, continuing the signal cascade. Lastly, nucleotides are important in energy metabolism. The best example of this is ATP and its production through glycolysis, the TCA cycle, and the electron transport chain. When ATP is converted to ADP, a large amount of usable energy is released in the form of a phosphate group. Transfer potential is the capacity a molecule has to donate a specific group. With ATP, ADP, and AMP, ATP releases the largest amount of energy, negative 30.5 kilojoules per mole. ADP releases the same amount, negative 30.5 kilojoules per mole, and AMP releases a smaller amount of energy, negative 14.2 kilojoules per mole. In Section 2, we are going to discuss purines and pyrimidines. Both molecules have cyclic rings that contain carbon and nitrogen. They have planar stacking, and they tautomerize into their oxo and amine groups. The difference is that purines contain 2 rings, while pyrimidines contain 1 ring. The most common purines are adenine and guanine, hence the mnemonic pure as gold, thinking of 2 gold rings. And the most common pyrimidines are cytosine, thymine, and uracil, hence the graphic of a pyramid, because there are 3 common pyrimidines and 3 points on a pyramid. Additionally, we must note that the keto-enol or amino-imino tautomerization of these nucleotides, the keto and amino forms will dominate at physiological pH because they have more resonance stabilization with their double bonds. And then I did want to show some less common purines and pyrimidines, namely 5-methylcytosine, 5-hydroxymethylcytosine, dimethylaminoadenine, 7-methylguanine, and pseudouridine. Next, we are going to talk about the different structures of nucleosides, which are a purine or pyrimidine with only a nitrogenous base, and nucleotides, which are a purine or pyrimidine with a nitrogenous base and a phosphate group. So in this image on the left, you can see the nucleoside by itself, and then it grows to a nucleotide. And so for each additional phosphate group, there is a name change. So adenosine monophosphate is AMP, adenosine diphosphate is ADP, and adenosine triphosphate is ATP, which represents how many phosphates there are on the molecule. And then in the right image, you may notice 2 different conformations of our nucleotide, syn and anti. These conformations differ about their N-glycosidic bond, which is between the sugar and the nitrogenous base. The anti-conformation dominates in nature because it rotates the nitrogenous base away from the rest of the molecule, minimizing unfavorable reactions and overall increasing stability. Lastly, I wanted to discuss some properties of nucleotides. Phosphate groups bear a negative charge at physiological pH, meaning that phosphoryl groups can serve as a proton donor or acceptor at certain pH values. At a pH of 7, most nucleotides absorb ultraviolet light at 260 nanometers, and, as mentioned earlier, nucleotides have high group transfer potential with a delta G of negative 30.5 kilojoules per mole and negative 14.2 kilojoules per mole. In Section 3, we are going to discuss analogs or modified versions of nucleotides that cause harm to the cell by resembling their counterparts closely enough to be accidentally incorporated. Here we can see some common analogs, a few that will be discussed more in depth later. Analogs are toxic to both transcription and translation. During transcription, if an analog is incorporated into genetic material in place of a nucleotide, it can cause disruptions in base pairing, premature termination, and polymerase stalling. During translation, analogs can cause changes in protein folding, which in turn affects protein function. There are two analogs I want to cover more in depth, cytarabine and azathioprim. Cytarabine is an analog of cytidine that is incorporated into DNA prior to cell division. This can cause apoptosis or trigger cell cycle checkpoints that can aid in targeting cancer cells. This analog has consequently been used in the development of new chemotherapy drugs. Azathioprim is a general analog of purines. This can cause suppression of immunologic rejection, making it valuable for targeting rapidly dividing cells like lymphocytes that mount immune responses against new organs. This analog ultimately slows the rejection process and is being used in patients who receive organ transplants. Finally, in section 4, I want to go over numbering. In this nucleotide, you can see that the nitrogenous base is located at carbon 1, the hydroxyl group is located at carbon 3, and the phosphate group is located at carbon 5. Overall, when naming cyclic carbons, the numbers go in order, running clockwise, and they're designated with this symbol right here, which is 1'. So we have 1', 2', 3', 4', and 5'. Now that we know how to number carbons, it's important that we know how to read genetic code. The phosphate backbone is formed through the elimination of a water molecule, or a condensation reaction, that links the 3' hydroxyl group to the 5' phosphoryl group. So you can see here the 3' and then the 5' phosphoryl group. DNA runs anti-parallel to itself, meaning that what is designated as the 5' strand is always on the left and the 3' strand is always on the right. And genetic code is always read 5' to 3'. So here on the left we can see 5' to 3', and here on the right we can see 5' to 3'. Lastly, I wanted to touch on the differences between DNA and RNA. DNA is regarded as more stable due to its lack of a hydroxyl group at the 3' carbon. This makes DNA better for a long-term storage of genetic code. RNA, however, has a hydroxyl group at the 3' carbon that acts as a nucleophile, making RNA much less stable. This is why RNA is used as a bridge between DNA and protein when going through transcription and translation, and has a shorter lifespan before degradation. These are the references used. You will find numbers on each picture throughout the slides that correspond to the sources here. And then I did want to go through some multiple choice questions just to practice what we've learned. So I'll just read them and then I'll kind of go through the answers. Question 1, which nucleotide plays a role in the regulation of the conversion of glucose to glucose 6-phosphate? CMP, ADP, ATP, or UDP? And if we think back to glycolysis and that first step, ADP is kind of what regulates hexokinase transferring the glucose to glucose 6-phosphate. Question 2. Secondary messengers regulate the ions that enter or exit the cell membrane, provide the genetic code for living organisms, oxidize NADH to NAD+, or bridge signals from outside the cell to inside the cell. And the answer is D, bridge signals from outside the cell to inside the cell. If you remember what we talked about earlier, secondary messengers are part of that signal cascade that bridges the extracellular receptors to the intracellular target proteins. Question 3. Which property of nucleotides is false? Nitrogenous bases form ionic bonds with each other. Phosphate groups bear a negative charge. At pH of 7, nucleotides absorb ultraviolet light at 260 nanometers. Nucleotides have high group transfer potential at delta G equals negative 30 kilojoules per mole. And the answer is A. Nitrogenous bases form ionic bonds with each other. That is false. They actually form hydrogen bonds with each other. Question 4. Which is false regarding analogs causing cellular harm? Analogs cause disruptions in base pairing, nuclear delocalization, premature termination, or polymerase stalling. The answer is B, nuclear delocalization. This was just never mentioned as one of the three types of ways analogs can cause harm to the cell. And then we have question 5. Which carbon is the hydroxyl group located on in a nucleotide? Carbon 1, carbon 2, carbon 3, or carbon 4? And the correct answer is C, carbon 3. If you look back on that graphic where we numbered clockwise, the hydroxyl group was attached to carbon 3.